Hi students, welcome to my channel Plant Science. Today we will talk about the plant embryology, in which we will discuss the unit development of male gametophyte, under which the topic discussed will be structure of Tetrasporangiate anther. Before starting with the structure of Tetrasporangiate anther, we will discuss few basic concepts. First, the plant embryology. Now, it is discussed as the study of structure and development of reproductive organs like stamens and carpel, male gametophyte and the development of male gametes, female gametophyte and the development of female gametes, process of fertilization, and at the last, process of embryogenesis. Now we will talk about the structure of a typical flower. Each flower is a reproductive organ developed on the plant. It is attached to the plant body by means of a stalk-like structure known as pedicel. Each pedicel has a flat end known as receptacle, on which all other floral organs are arranged. Now, the floral organs are basically divided into two broad categories. The first one is accessory whorls and the second one is essential whorls. Now, accessory whorls are known as accessory because they do not participate in the process of actual sexual reproduction, but they are helpful for the process of reproduction. Now, accessory whorls are further divided into two categories. The first is calyx and second one is corolla. Now, calyx has individual member known as sepal. These are usually green in color and they are protective in function. Now, the second corolla, these are mostly brightly colored and each individual member of corolla is known as petal. Both calyx and corolla are involved in the protection of essential whorls like androecium and gynoecium present inside them. Now, we will talk about the second part, essential whorls. Essential whorls are again categorized into two categories as androecium and second, gynoecium. Androecium is a male reproductive organ and each individual member of androecium is known as stamen. Now here, if we put the stamen, each stamen is divided into two parts, the anther and the filament. Now, anther consists of pollen grains at the maturity, while filament is involved in the attachment of the anther towards receptacle. Now, the second part, gynoecium. Gynoecium is a female reproductive organ of the flower. Now, each gynoecium is consisting of individual member known as carpel. Each carpel is divided into three parts. The basal part as ovary, the middle part as style, and the distal part as stigma. Now this entire composition of ovary, style, and stigma represents a single carpel. Here we will discuss about the outline of process of sexual reproduction in plants. Now first we will start with the anther of the stamen. Now as I have discussed previously, the anther is a reproductive part of the androecium or stamen. Now each anther at the younger stage consists of microspore mother cells. Each microspore mother cell is diploid in nature, which undergo meiotic division or meiosis to form microspores or pollen grains which are haploid. This process of formation of microspores or pollen grains from diploid microspore mother cell is known as microsporogenesis. Now, each microspore or pollen grain further metamorphose into the male gametophyte, which will develop the respective male gametes. On the other hand, in case of female reproductive organs, the ovule located within the ovary of the carpel consists of a single megaspore mother cell. Megaspore mother cell, which is diploid, undergo meiosis to form the megaspores, which are haploid. This process of formation of megaspores is known as Megasporogenesis. Megaspores develops into the female gametophyte that will produce female gamete. Now, the male gamete and female gamete undergo the process of fusion known as fertilization that will result in the formation of a diploid zygote. Now, each zygote undergo the further successive developmental stages to form an embryo. This formation of embryo is known as embryogenesis. Now we will discuss the structure of a typical stamen. As you can see in the diagram, each stamen is divided into three basic parts. The first is anther, second is filament, and the third is connective. Now, the anther represents the fertile part of the stamen, which means it consists of the sporogenous tissue that will develop into the functional pollen grains. Each anther is further divided into the compartment-like structures known as anther lobes. Number of anther lobes are usually 2. Each anther is connected to the receptacle by means of the filament. Role of the filament is to provide the nutrition to the developing anther and the developing pollen grains. Now, the third part, connective, is involved in connecting the two anther lobes together, but also it is considered as the extension of filament that is consisting of vascular tissue and also providing nutrition to the developing pollen grains or the sporogenous tissue. If we discuss the anatomy of a typical anther, then each anther shows two anther lobes, while each anther lobe consisting of two chambers, also known as pollen sacs or microsporangium. Each microsporangium or pollen sac consisting of sporogenous tissues that will develop into the pollen grains at the maturity of an anther. Each such anther with 4 pollen sacs is therefore known as Tetrasporangiate anther. Now, it consisting of following parts as epidermis, endothecium, middle layer, tapetum, and the sporogenous tissues. Now, each part we will discuss in detail in the following part. The outermost layer of cells is known as epidermis. It consists of the lignified or cutinized cells which are thin and flat. These are protective in function and involved in the protection of the entire anther. It covers all the 4 anther lobes as a common layer. Second layer next to the epidermis is known as endothecium. Endothecium is single layer of cells, each cell consisting of the fibrous thickenings of callus. The cells of endothecium are hygroscopic in nature, which absorbs the moisture from the surrounding, and therefore they are helpful for the dehiscence or the burst of the anther at the maturity. Dehiscence results in the release of pollen grains in the surrounding outside the anther. Epidermis and endothecium surrounds the entire anther, while the third layer, known as middle layer, surrounds each pollen sac or microsporangium separately. Now, this middle layer consisting of 1 to 3 layers of the parenchymatous cells, each cell is flat and thin-walled. Now, these cells are ephemerals, that means they will degenerate at the maturity, providing the nutrition to the developing sporogenous tissues. Tapetum represents the innermost layer of the anther. It surrounds the entire microsporangium or pollen sac completely. The cells of the tapetum are larger in size with dense cytoplasm and prominent nuclei. Some of the cells may be multinucleated. Now, their primary role is to provide the nutrition to the sporogenous tissue and developing microspores or pollen grains. Tapetum are of two kinds based on the type of nutrition mechanism. The first is amoeboid tapetum and the second one is secretory tapetum. Tapetum is marked here by the dotted line, as you can see in the diagram. The portion between two dotted lines represents the single layer tapetum, while the inner mass represents the microspore mother cells or the sporogenous tissues. The chamber within the anther lobe is now known as the pollen sac or microsporangium. Each microsporangium consists of diploid sporogenous tissues. Initially, these diploid sporogenous tissues divide by mitosis so that the number is increased to ensure the production of large number of pollen grains. Initially, the sporogenous tissues will metamorphose and develops into the microspore mother cells, also known as pollen mother cells. These pollen mother cells are diploid in nature. Each of such pollen mother cell divides meiotically or by meiosis to form the set of 4 haploid spores. Such set of 4 haploid spores is known as microspore tetrad, and the process of formation of such haploid spores from the diploid pollen mother cell is known as microsporogenesis. There is a formation of a tetrad which consisting of 4 haploid spores togetherly. Later on, these haploid spores will separate from each other and then they will develop into the single pollen grain. Number of pollen grains formed depends on the number of pollen mother cells present in the pollen sac or in the microsporangium. Means each pollen mother cell give rise to 4 haploid pollen grains at the maturity. You can see in the diagram that the initial spore mother cells are being replaced by the matured pollen grains in the microsporangium. Such matured anther with the developed pollen grains is now ready for the dehiscence. This is all about the structure of typical tetrasporangiate anther in this video. In my next video, I will discuss the structure of typical pollen grain and the development of male gametophyte. Thank you for watching. Like and subscribe to the channel for more videos.